Before we plunge into interesting things, the College of Medicine is moving us Friday. And the class will meet in Odd 1, which is Beisner, BSB. Sorry, BSB. We've been there before. This time we know we're loaded with microphones and with recording devices. So hopefully there won't be any difficulties there. I'll fix the lowercase b later. So meet us over there on Friday. Let's go ahead with our class things. Are there any questions from Monday's discussion you want to touch on before we move on to today's? There were a few slides at the end, and I'm going to let you finish reviewing those on your own. It's only a 3-session review. If we start to get behind, we can't catch up. So, reminding you that the final exam will be a week from yesterday. We are in 2 locations. It's important to bring some kind of ID with you because we really don't have spare room. And people without ID will be asked to wait to be seated until we know we have room for you. A through M here, N through Z over there. So try to be ready. Not my favorite solution, but I haven't yet gotten them to give me a better one. So, okay. So, we were talking about protein structure, and we are edging up onto myoglobin and hemoglobin, and the fact that both myoglobin and hemoglobin have primary structure, which is their sequence. They have secondary structures, which in the case of myoglobin and hemoglobin is a number of alpha helices. And they both have very similar tertiary structures, despite the fact that their sequences are very different. Hemoglobin, because it has 4 polypeptide chains, it's an alpha-beta dimer, has quaternary structure, which creates in the center of the tetramer a cavity that can be occupied by BPG or bisphosphoglycerate. And that's in addition to the binding sites. In each individual subunit, each subunit can bind 1 oxygen in its heme pockets. And the influence of BPG is very important because BPG has on it a lot of negative charge. That central cavity in the middle of the tetramer has positive charges. And so what it does is it helps stabilize hemoglobin in the deoxy state and makes it easier for oxygen to come off when BPG is around. And quite how dramatic it can be, we can see it by comparing this graph. And again, the half saturation point is a measure of affinity. So this is half saturated around here. This is half saturated around here. And what it shows us is that it's without BPG around, hemoglobin will not release very much of its oxygen in the tissues, whereas with BPG, it'll do a better job of that. This trick of controlling oxygen release by hemoglobin affinity actually goes so far as the fetus inside has different hemoglobin that allows it to have pull oxygen away from the mom's circulation. So now let's move on to one of the ways in which proteins are used that's a very important part of the life of cells, and that is the cytoskeleton. Cytoskeleton is principally in eukaryotes. There are some analogs in prokaryotes, but we're not going to discuss those so much. There are 3 major filament systems in the cytoskeleton. There are the microfilaments, and I'm not quite sure why I made this one so fancy. Microfilaments are going to be abundant in a number of tissues that need to either contract, like muscles, or platelets actually contract, or have short rigid projections, like microvilli in the gut. A second major class are the microtubules, and we find that the microtubules are very abundant in cilia, structures that are involved with making water move. We have those in our lungs and in the reproductive tract. And neurons that are very long, skinny cells and need all the help they can get. And then intermediate filaments, which are very abundant in epithelia of various kinds, so skin and hair, the gut, but also in neurons, again because neurons are very long, skinny cells that need all the structural help they can get. Oops, I'm sorry. Didn't mean to do that. There we go. Oh. There's a disadvantage to that. All right. This is a picture showing an epithelial layer that has the microfilaments highlighted. We've mentioned their abundance in the gut and in muscle. Microfilaments are going to be abundant when cells are holding on to something, adhering to something, either to each other or to a surface, contracting like muscles or being stiff. The components of microfilaments, the things that are the building blocks, are called actin, and actin is a monomeric protein. Sorry. Okay. The thickness of the actin filament is 7 nanometers. That's why they're the thin filaments, or the microfilaments. If we look at microtubules, we can see, again, we've mentioned their abundance in the cell types. Cells that swim will have a lot of microtubules. So in us, that would be sperm. In protozoa, a number of places we see cilia for swimming. They're involved in stiffness in parts of the inner ear, and especially an important role in mitosis and cell division. The microtubules provide a framework for the chromosomes at cell division. And this is a picture of a HeLa cell, and it's, down here I want to show you the microtubules. Here you can see the microtubules radiating throughout the cytoplasm. Microtubules can also be railroad tracks for vesicles and stuff. They're made up of a protein tubulin, no surprise. There's two flavors of tubulin in any microtubule, alpha and beta. So it's a heterodimeric rather than a monomeric thing like microtubules. The intermediate filaments, we've mentioned where they're abundant. It's especially important for tissue strength, for attachment, for being able to resist strains on it, so that the parts of your skin that undergo the greatest mechanical exertion are going to have the densest intermediate filaments. Skin up on your face, which undergoes almost no mechanical stress, has some, but not as much. The stuff on the balls of your feet has a lot more. One of the best sources of the attachment things for this that a friend of mine isolated was from cow noses, because they have to burrow through the grass. Intermediate filaments are much more heterogeneous as proteins than the other two classes. There are 5 families of them. We're not going to try to pin down each of those 5 families in detail. They get their name of intermediate filaments because their filaments are 10 nanometers thick, so they're between the micro thin filaments and the thick filaments, which are the myosin we'll see in a little bit. And this diagram captures fairly nicely, I think, the key features of the 3 cytoskeletal filament systems. Actin filaments here are the thinnest of them. You can see it's a thin filament. It's made up of a monomer in a helical packing of globular proteins. Here's your monomeric G protein. When it makes a filament, it becomes F-actin, G-actin, F-actin. Here you can see the thing is a tubule with a hollow center. It's clearly quite a bit bigger than the thin filaments. And you can see here how the heterodimers are stacked head to tail to make up the walls of the microtubule. And then by comparison, the intermediate filaments are coiled coils. You can see how they've been packing in here peptide chains side by side and then staggered relative to each other. Both the actin filaments and the microtubules have a polarity to them. There's a difference between one end and the other. You'd have an alpha tubulin at one end of this and a beta tubulin at the other. In the case of the G-actin fitting into here, it fits in one orientation and not the other. So that these two structures are polar in the sense that they have differences between their two ends. And we'll see that in a second. By contrast, the intermediate filaments are symmetrical from one end to the other. One interesting thing about the intermediate filaments is it's the first place that we encountered a very important structure called a coiled coil. And because at 7 subunit repeats, there were hydrophobic side chains, that allows an alpha helix to coil around another alpha helix, and that's what the coiled coil is. You can see that in a backbone picture here. It's held together by hydrophobic interactions between there, but those coiled coils recur in a number of contexts, and we'll point them out again as we run into them, but it's a common structural motif. Intermediate filaments, by contrast to either tubulin or actin, do not bind any nucleotides. They don't bind nucleotides, they don't hydrolyze nucleotides. So they're simpler in that respect than the other two classes. And I've just mentioned that this is a common way to build dimers, of having a coiled coil with a heptad repeat. So, okay, and that's just showing you that in more detail. There we go. Oops. Let me just page up once. No, that's it. Okay, there we go. All right. So what we've talked about so far with the cytoskeleton proteins are things whose principal roles are structural one way or the other, of making motion or resisting tension or generating tension or being stiff. They're only incidentally enzymes in that actin and tubulin can hydrolyze nucleotides. But then there's the whole vast universe of proteins whose reason for existing is to be enzymes. And so by taking a little peek at chymotrypsin, we can see some common features of enzymes in general. This is a digestive enzyme. Its sole role is to add a water molecule across that peptide bond, the semi-planar character there that makes the peptide backbone have its characteristics. And it vastly accelerates the reaction. So it's 10 to the 11th times faster with it than without. So this is going to be kind of a model for seeing certain features of enzymes that occur again and again. It has a region on it here that's going to be the place where the hydrolysis happens, and we call that the active site. So there's always going to be part of the enzyme that's most involved in catalysis directly. One of the things that this enzyme does, like many enzymes, is it allows the cell to do this under mild conditions. You can break down proteins very easily by putting in hydrochloric acid or sodium hydroxide, but chymotrypsin can do it near neutrality. By looking at it and its siblings, we can see some of the roots of specificity, how enzymes can select one thing over another. And what is it that enzymes do? In order to know what enzymes do, we have to look at reactions and their free energies. So if you start off with some reactants of some kind, and you allow the reaction to proceed to its products, you can really have only two situations. You can have one in which there's a negative free energy change between the products and the reactants, or a positive free energy change, or none, obviously. But between the reactants and the products, there is a hill of the transition state, the configuration that the reactants have to go through to get to the other side. So there's actually 3 energy spots to be paying attention to: the reactants, the products, and the top of that hill, the transition state intermediate. Now, a negative delta G will tell you something about how spontaneous a reaction is. If it's a negative delta G, it can happen without energy being put in. If it's a large negative delta G, it will probably go largely to completion in the absence of other circumstances. But none of that, the size of the delta G of the reaction, tells you nothing about the rate of the reaction. The rate depends on the height of this hill. Since the reactants have to get over that hill to get down to being products, the higher the hill, the less often there's going to be a random input of energy from the environment, the less often the reaction will happen. So you could have a very large delta G, and if the hill were high enough, it'd never go there. Essentially never. Not while we're alive. People talk about, half-times of reaction of 10,000 years, and I'm not going to stay around to see what happens. So what do enzymes do? Here we have, again, a reaction coordinate. We have reactants here. We have products. This is a reaction with a negative delta G, so that it's going to have some preference for the products over the reactants at equilibrium. And in the higher line, the red one, this is in the absence of any catalyst. And the magic of an enzyme or any catalyst is that it allows for a new way of getting there with a different transition state that is of a lower free energy difference than the previous transition state. So it lowers the energy of the transition state. Since this hill is lower, it's more often there's going to more often be enough energy to go over it. The rate will be accelerated. The rate at which it goes forward will be increased. The final extent will not change at all. There's been no change to the delta G of the reaction. Therefore, there is no change to the overall extent. Any questions on this point before we go on? Okay. So what enzymes do is they create peculiar, specific chemical conditions. And some of them do such peculiar things that they need something other than just the side chains of amino acids, and we call those other somethings cofactors. Many of the vitamins are cofactors to enzymes. Broadly speaking, there are three major mechanisms. There are some others that get more exotic, but these cover a lot of areas. Acid-base catalysis. I mentioned you use hydrochloric acid to break down proteins. That would be acid-base catalysis. There are covalent mechanisms where the transition state is actually connected to the enzyme itself with a covalent bond. And in some cases, a metal ion plays its little magic tricks. And this is one of the reasons that trace metals are important in our diet. Chymotrypsin has been a classic for looking at enzyme mechanisms as a model for that for years because it captures several of them at once. It's an example of acid-base catalysis, and it's an example of covalent catalysis. And this little trio of side chains here of aspartate, histidine, serine has been called the catalytic triad because these three side chains specifically account for most of the catalytic activity of chymotrypsin and, by extension, other members of its family. You can see how these are coordinated through hydrogen bonds to each other and placed in a very specific geometry relative to each other. Well, if they're that important, what does the rest of the enzyme do? Early on, people thought maybe it was like a lock and a key, which was a good metaphor to the extent that it tried to capture the basis for specificity, and enzymes need to be specific. It was actually a problem that if the substrates are tightly bound to the enzyme, you actually would make it harder to get over the hill rather than easier. And so some of the features that we look at now are like this. Proximity, holding the substrates closer to each other, in effect raising the concentration. Orientation, holding them pointing in a particular way so they can fit just so. Induced fit, a surprising number of enzymes change their shape when the substrates bind. So it's like a lock that changes its shape when you put the key in. There's electrostatic contributions to it where an electrical field can influence the ability of protons to come on or go off. The enzyme will have a special site that's snug enough for the reactants so water can't get in there unless that's one of the reactants. And since in the absence of an enzyme, water is overwhelmingly present, this alone can help influence what reactions do and don't happen. This family of proteases is a nice example of evolution in proteins and the things we can learn about evolution and the things we can learn about proteins by comparing them. Here we've overlain three proteases that are related to each other, chymotrypsin, trypsin, and elastase. You can see that they have very similar tertiary structures, that the overall packing in three-dimensional space is very similar. Their primary structures or their sequences are quite different. And we saw that also with myoglobin and hemoglobin. Myoglobin and hemoglobin have very similar secondary structures, very similar tertiary structures. Their primary sequences are not nearly so similar. However, in all three of these guys, this catalytic triad here in the middle is perfectly conserved. The as, pis, ser, those three catalytic residues are exactly there. The surface residues, whose only job is to make sure that it stays soluble, vary much more. So that points us at the idea that you need that group to have catalysis. What can we learn about specificity? Because accelerating reactions is fine, but you want to accelerate the right reactions. Well, chymotrypsin is picking up on large hydrophobic residues. Trypsin cuts after lysine and arginine, so the positive charges. And elastase likes to cut after little things that are hydrophobic, like alanine, glycine, valine. And if we look in the active site, here they're pointing out what they call the scissile bond, the bond that has to be broken. So it's up about there in each of these little diagrams. The side chains then stick down, and there's a pocket. And the shape and the characteristics of that pocket determine what things can fit into it, and therefore what things the enzyme can do anything to. So for chymotrypsin, there's a deep pocket that's hydrophobic. For trypsin, still pretty deep, but it has a little negative charge at the bottom. And so now it's going to select out the positively charged lysine and arginine. And for elastase, side chains are sticking into there to make it a shallow hydrophobic pocket. And since it's shallow, it only allows small hydrophobic things in. So the active site as a whole is a combination of the catalytic residues that can actually catalyze reactions and specificity pockets that contribute to binding the substrate, so you can select the substrate that you're going to do something to. And it's helpful to be able to think at least a little bit about how fast enzymes do things. And that whole realm of thinking is called kinetics. And if we start under conditions where you have a great deal more substrate, the reactant, than we have enzyme, if we add more enzyme, the reaction will go faster. So when the substrate is in vast excess, the enzyme will determine the rate of the reaction. But if we pick a specific concentration of enzyme, hold the enzyme fixed, and now we vary the substrate concentration, we see something slightly different. Initial increase in reaction velocity with more substrate, but adding more substrate doesn't keep on giving us a greater reaction rate. Eventually it tapers off, or it continues to taper off. We call that shape of a curve a hydrophobic curve. And what's, sorry, a hyperbolic curve, excuse me. I'm not completely well yet. And what the hyperbolic curve is showing us is that that enzyme can get saturated, that there's some fixed rate at which the enzyme turns over to product, and that if every enzyme is saturated, that's as fast as everything can go. And we interpret this as telling us that you can start from a free enzyme and substrate to make an enzyme-substrate complex, and when it's catalyzed the reaction, releases it, you now have free enzyme and product. You should probably recognize the shape of that curve from the myoglobin binding curve, for example. And so when we think about enzymes, there are two characteristics we can think about. One is how fast that enzyme goes when it's completely occupied, the maximal velocity when it's completely saturated. But the second property is to ask how much substrate does it take to saturate it. Now, since the curve is hyperbolic, it's hard to get a number for completely saturated. That's off there in, you know, another building. And so like so many things, we ask, how about half saturation? And so the amount of substrate that gets an enzyme to go at half of its maximal velocity is the Michaelis constant. It's the concentration of substrate at which the enzyme is going at half of its maximal rate. And that is a measure of how well it can select the substrate. It's a good approximation for the affinity of the enzyme, especially when the forward reaction-making product is the limiting aspect of it. One thing that in English is a little confusing is that affinity and Km go in opposite directions. If you talk about a large Km, a lot of substrate to half saturate it, we would usually say that's a low affinity. If you say something has a high affinity for something, it doesn't take much to saturate it. That would be a small Km. So the words go in opposite directions. That's a little bit of a drawback to them. So life is rarely as simple as that reaction. It's quite common for reactions to include many components, and this is just one example of a true reaction, real-life reaction. You may happen to recognize it as coming from the pentose phosphate pathway or the Calvin cycle, but that's not important. What is important is look at all those arrows. Every one of those arrows has rate constants. But we can still speak of a catalytic rate going from here to there as an overall rate of catalysis, even though each one of those association-dissociation events has its own rate constants. And then, as if life weren't complicated enough, there are enzymes that don't have hyperbolic kinetics. So, for example, some enzymes look like this, where as you change the substrate concentration, there's a small increase in velocity rising up to its half maximal, and then and then it tapers off, or what we call sigmoidal kinetics. Most commonly, sigmoidal kinetics will occur when there's some kind of cooperative behavior. So sigmoidal characteristics are generally found for multimeric enzymes. We saw things like this in the oxygen binding for hemoglobin, for example, which has sigmoidal oxygen binding. It also is typically a consequence of allostery, of having one place on the enzyme affect activity somewhere else. And that's all that allostery means, other place, except they say it in Greek. We can still think about things like a Vmax of how fast an enzyme goes when saturated, and a Km, what it takes to half saturate it, or as an indirect measure of affinity. But how we get those numbers is a little harder to talk about. We don't need to worry about that. Now, both inside of our cells and in life practice, like in the fields or in the clinics, we don't always want enzymes to do things. We want to be able to inhibit them. So enzyme inhibition is almost as important as enzyme activity. And I'm just going to, in this review, touch on two kinds of it. I don't want to go over all the different kinds. One striking kind is irreversible. So we looked about the example where there's a substrate that mimics a peptide and binds to the thing and reacts with it, but stays bound. It's become covalently attached. Sometimes we call this a suicide substrate. It kills itself in the process. And that is a dead enzyme. It's not going to ever do anything else again. It often reacts at the active site, but you can also get irreversible inhibition elsewhere on the protein. Sometimes you want to be able to do something less drastic, or sometimes you can't find a magic suicide substrate, in which case competitive inhibition is a common next tool or step. If the substrate fits into the active site in one particular way, there may be other molecules that are similar enough in their shape that they can bind at the active site, but they don't do anything there. They come in and they go away. And that's a very common characteristic. But since the enzyme is spending some of its time occupied by the inhibitor, it can't do as much to the substrate as it would in the absence of the inhibitor. And this is what that looks like kinetically. If we look at an enzyme that has this kind of kinetics in the absence of an inhibitor with a competitive inhibitor that's simply binding at the active site and going away, what we see is that it takes more of the real substrate to get the same half maximal velocity. You have to have more molecules of substrate around to have the odds that that's what's going to be bound instead of the inhibitor. And so it appears to change the Km. It appears to change the affinity to make it a weaker affinity. But there's no reason to expect it to change the Vmax in a pure competitive inhibitor. We often see examples of allosteric inhibition, where an enzyme can be controlled by other metabolites. Sometimes that is simply being controlled by its own product, which would just be ordinary feedback inhibition because there's a homology. I mean, it obviously could be at the binding site because it just came from the binding site. Or some other metabolite, as in this example, where phosphoenolpyruvate, which is well downstream from phosphofructokinase, comes back and can inhibit the enzyme. And this allows metabolic pathways to have coordination and control. In this case, phosphoenolpyruvate is not one of the substrates, and so it's acting allosterically. It's also worth knowing you can have allosteric stimulation, that it's not just turning things down. In some cases, you turn things up allosterically. And that would be in a little more detail here. This is the kinetics for that diagram on the previous page. Here's your phosphofructokinase, a nice hyperbolic curve in the absence of phosphoenolpyruvate. Very, very high affinity. Takes very little of the substrate to bind to it. And when you add phosphoenolpyruvate, which is that downstream product, you can think of it as meaning, if you like, we've got the product, don't give me more. And now you can see a very great change in the affinity for it. It takes much more to half saturate it. But the actual Vmax is unaffected, sort of on the principle of, well, if it's around, I guess at some point I have to do something with it, you know. We actually call this state a tense state in analogy to hemoglobin. And interestingly, ADP, which would be a marker for low energy state, helps keep it in a more relaxed state. And it, of course, would be a good marker for needing glycolysis to go forward. So. And then we come to another way that proteins can be used, or if you want to think about it, we come to the other structural component of living systems, and we need to think about membranes a bit. Membranes are the boundary that give a cell an inside and an outside. And they're basically mixtures of lipids and proteins, a certain amount of sugar on the outside. We're going to see that they are bilayers of lipids. We talked about that a little earlier with the hydrophobic thing, that one thin layer of lipids here and another thin layer of lipids there, put their hydrophobic bits together, and their hydrophilic parts on the other side. So the lipids contribute the bilayer aspect. The proteins do most of the rest of the functions. So the lipids can seal things, but the proteins get interesting. Lipid bilayers are not covalent structures. They just spontaneously arise when there are enough of the right kinds of lipids around. They're cooperative. And in most living systems, they're asymmetric. That is, they don't have the same exact mixture of lipids on the two faces. People have tended to talk about the fluid mosaic for membranes, and I tried to emphasize that a more accurate one would be a solid mosaic one in terms of the proteins, at least. You can call the lipids fluid mosaic, if you like, because there's some variety there, and they're certainly fluid. But the proteins don't really behave that way, and we'll mention that a little more. Almost all membranes in living systems are electrically polarized, that there's an electrical charge across them. That's one of the signs of life. Just as we look for being electrical activity in the brain in a human being, a living cell will have an electrical difference across it, and when it loses that electrical difference, in many cases, that's a dead cell. Here are a couple of examples of membrane lipids with their very hydrophobic tails here and their polar hydrophilic heads. And here is a cholesterol, which somebody may have noticed by now. I flipped in Photoshop because I wanted to have the polar part on the same side of the page. So that's that little itty-bitty tiny bit of polar character there. All the rest of that is hydrophobic. So the hydrophobic interiors create the sealing, like the joint grease on a valve. The proteins then can either actually transfer molecules, actual physical things, across that sealed layer, or just transfer information. We call proteins that completely span the membrane, clear from one to the other, integral membrane proteins, ones that are on the edge of the membrane and not interior to it, peripheral membrane proteins. Even for integral membrane proteins, domains here and here are going to have hydrophilic side chains exposed to the watery parts. The side chains in contact with the lipid bilayer will usually be hydrophobic. Here's some of the different kinds of things membrane proteins do. They can be channels for passive movement. They can be pumps to actually create gradients. They can be receptors that are specific for ligands and therefore read information from the environment. There are even some enzymes that live on the membrane or in the membrane to accomplish certain jobs. So if we look at a membrane, we see that the lipid components move rapidly, often in the general range of the rates that we would expect for diffusion. The protein components usually move slowly, typically in the order of orders of magnitude less than expected, or immobile on any time scale that cells care about. And in this case, we're always talking about movement in the plane or parallel to the plane of the membrane. Going across the membrane from one leaflet to the other, flip-flopping, is rare. It doesn't happen spontaneously at a very significant rate. There are enzymes that will flip lipids from one side to the other, and that's how cells can have differences between those two layers. The reason that the membrane proteins don't behave like the lipids is because cells have cytoskeletons, and you can see here the artist's loose attempt to try to show you that the membrane proteins are not just icebergs in a lipid sea, as they're often called, but in fact they're anchored quite nicely to cytoskeletal elements. And this is one place where I contradict the book. And you're welcome to try to talk me out of it if you want to. So moving things across membranes can happen passively. If there's a gradient, things will just flow downhill. They can happen actively. So the sodium ATPase, for example, uses ATP to do that. These channels that are just simply letting things go passively are specific, and they're a good way for ions to communicate things. There are further special structures called gap junctions for larger chemicals to get across. Whether or not something moves through a membrane is going to depend on its general characteristics and whether there's an energy source. So there are some nonpolar hydrophobic things that will simply diffuse through passively, like a steroid. Nothing carries steroids through a membrane. That's simple diffusion. You have facilitated diffusion, where you have a channel and things that would not be able to go through that hydrophobic interior can pass through the interior of the channel. And then active transport involves using energy to create a gradient. And this is just a picture of the gap junctions that connect cells, like the cells in your heart are connected by gap junctions, and good thing too. They will permit ions to go through, but they'll also let small molecules pass as well. Probably the most dramatic thing that membranes do is they're the location at which the majority of signaling happens, and that's going to happen through membrane receptors. You need to have a signal, which might be a hormone or a small molecule or whatever. There's some kind of reception at the level of a membrane receptor, some means usually to amplify it. The steroid hormones are exceptions to that, of course. And then further amplification there. Commonly we see protein phosphorylation used. It's not the only means, but it's a common one. And there's always some way to turn things off. There's one very large family of proteins called the 7TM receptors with 7 transmembrane domains. And this is just showing you the similarities for 2 of those strategies of oh, sorry. Hate to think of someone at home on their computer having to hear that call. So this is showing you the similarities down through to the point of, in this case, using G proteins, and that those G proteins can activate either adenyl cyclases or phospholipases. If they activated adenyl cyclases, you're going to get cyclic AMP. That cyclic AMP can activate various things like protein kinase A, which then are going to target serines or threonines of specific proteins. If it had gone down the other fork of this, that phospholipase C is for calcium. It will increase the levels of two components which also raise calcium concentrations, which will then activate protein kinase C, which stands for calcium. And it also has serines and threonines on targets, usually different targets. These are just some of the examples of systems that get activated that way. Glycogen metabolism, a variety of genes, synaptic transmission, your ability to remember that you saw something before. Some of that's dependent on this pathway. In some cases, receptors can end up responding to the ligand. So here we have EGF as an example. Once it's bound to its receptor, it can find a partner that's also been occupied and then phosphorylate each other, for example. It's another strategy that gets used. And we can prove that the external domain isn't really giving specific information because we can put a different domain out here plugged onto this end here, and it will turn things on as if it had been EGF. So the specificity inside the cell comes from this part, not from up there. Shifting gears to metabolism a bit. We need to take energy out of the environment, reducing power. We need to build metabolites and macromolecules. We use pathways that have multiple steps that are going to connect to things. We oxidize fuels to get that energy. And we see certain motifs over and over again. So if we take fuels and oxidize them, we're going to end up with carbon dioxide and energy of one kind or another. That whole group of things is catabolism. Glycolysis is obviously one aspect of that. Oxidative phosphorylation, you might want to fill in what are the other catabolic pathways. Once we have usable energy and small molecules, we can make bigger things. We call that anabolism, like anabolic steroids that build up. Gluconeogenesis, glycogen synthesis. Again, you might want to fill in what are some of the other anabolic pathways. There are some very important pathways that can both break things down and oxidize them, and in related modes, build things up and renew things. And those things get called amphibolic. The citric acid cycle and pentose phosphate are two obvious ones, and others too. We've met along the ways a variety of compounds with high phosphoryl transfer potential or high energy bonds. ATP is sort of the hallmark of them. You could put a G up there. This energy part would be just the same. You can sort of look and see why phosphoenolpyruvate would be a high energy compound, why 1,3-bisphosphoglycerate might be as well. So these are all of them key compounds. But energy isn't the only thing that we need to be able to carry around. We need to carry carbon fragments. The various CoA things like acetyl-CoA, acyl-CoA, succinyl-CoA are carbon fragments that are carried around. We need to carry electrons, and there are a number of electron carriers there. We need to be able to carry sugars, UDP sugars, and so on. So that lets you refresh those. This is sort of the grand overview of glycolysis. That you're going to feed in, break down polysaccharides into the monosaccharides, glucose, other sugars. Glycolysis is going to be able to break that down. Under aerobic conditions, we'll get it to acetyl-CoA, feed it to the citric acid cycle where 2 carbon dioxides come off. And the electrons that we're going to strip out will be fed into the oxidative phosphorylation. Oxygen will be the electron acceptor to come off as water. And the net effect is ATP, because as those electrons go through, we create a proton gradient, and that lets us make ATP. And the next several slides attempt to sort of summarize major pathways for you. I can see we're getting a little bit late on time, but I won't necessarily go through all of them. But what they've done is to try to organize for you related pathways in terms of their similarities and differences. And this may be a way to refresh your memories on them. Glycolysis, and gluconeogenesis are both involved with sugar. You can hear that in the gluc beginnings, breaking down, making sugar. They both involve the 3 and 6 carbon sugars. They're both under control. They're both cytoplasmic. But the glycolysis will be the catabolic one. Every tissue has to have it, and it's going to release available energy in the form of ATP or acetyl-CoA. Fructose 2,6-phosphate and AMP activate it because those are either a marker for low energy status or regulated metabolite. Gluconeogenesis, by contrast, is the anabolic version of it. It's going to build sugars up. It's not everywhere, just a few places. It's going to cost energy, ATP and GTP. And now fructose 2,6-phosphate and AMP inhibit it, and they will share many reactions but bypass 3 that are going to cost energy to go back uphill. I don't know if people find it valuable to go through these one-on-one together. I'm certainly happy to go on doing that. But I think you can see how they're organized to help you review your thoughts. All right?